Now, in the very end of Chapter 3, you will find a little bit of information about some relatively new advances in molecular cytogenetics, that is, some relatively new ways of studying chromosomes. These two techniques are known as FISH, fluorescence in situ hybridization, and spectral karyotyping, or chromosome painting. These two techniques have both been derived from the Human Genome Project. And you're probably aware that we've been working for approximately the past 20 years or so to determine the complete DNA sequence of the human. And we're essentially finished with that now. There are still a few small areas that have not been completed, but we now know almost our complete DNA sequence. And because we know our complete DNA sequence, we now know the DNA sequence of almost all of our genes, both normal and many mutant copies of genes as well. And with the technology that has been developed from these experiments, we have instruments called oligonucleotide synthesizers, or gene machines, that will automatically synthesize DNA for us. So we can go to the lab and type into the computer part of our gene machine the DNA-based sequence of any gene that we want, push the start button, go read a book for a few hours, and when we come back at the end of the day, the machine has automatically made for us a single-stranded piece of DNA corresponding to the sequence, the base sequence of any gene that we want. And that single-stranded piece of DNA can then be used as a probe to base pair with or bind to the naturally occurring genes on our chromosomes. And furthermore, we can tell that gene machine to attach a fluorescently labeled molecule to that single-stranded DNA probe so that we will be able to find where it has bound by shining an ultraviolet light on the chromosomes after allowing the probe to bind. So that kind of situation is used in a technique called fluorescence in situ hybridization, or FISH. Here is the kind of diagram that you might possibly see on your exam related to FISH. In this example, what we're illustrating here is that someone has made a FISH probe to some particular gene on chromosome number 10. They have exposed the chromosomes to this probe, allowed the probe to base pair or bind to the naturally occurring gene. We shine an ultraviolet light on those chromosomes and take a picture. And the idea is that fluorescent probe will, under the ultraviolet light, will give us a red spot or blue or green or yellow, depending on what the particular label was. It will give us a red spot, for example, at the position where the probe bound to the naturally occurring chromosome. And so the position of this red spot here would identify the locus for that particular gene on chromosome number 10. That is, this would tell us exactly that. First of all, it would tell us that this is chromosome number 10, and it would tell us exactly where that gene is located on chromosome number 10. Now, just suppose that this diagram represents pairs of chromosomes. What's wrong with this picture? If you saw this on your exam, what is wrong with this picture? Well, if these are supposed to be the two copies of chromosome 10 from an individual, both copies should have a red spot. So the idea is that if we only get a red spot on one of the copies of chromosome 10, that's most likely to mean that there is a deletion on the other copy of chromosome 10. The probe was not able to bind because there is a deletion on this copy of chromosome 10, a deletion that might be too small to see under the microscope. So this is one of the ways that FISH probes are used to identify small deletions in chromosomes. The other major technique described in this chapter is spectral karyotyping or chromosome painting. In this technique, people have developed a family of probes labeled with different colors, different fluorescent colors, and the cell is broken open, and all of the chromosomes are exposed to this mixture of different colored probes. Eventually, an ultraviolet light is shined on the mixture, and we take a picture and let a computer analyze the picture, and the computer will prepare for us a diagram like this, in which each individual chromosome is painted its own specific color. This is a much easier way to identify particular chromosomes compared with the older banding patterns that were previously used. Because of the distinctive colors, it's very easy to tell one chromosome from another. I think this picture would make a great test question for the Step 1 exam. If you saw this picture on your exam, what's wrong with this picture? Well, this represents a reciprocal translocation. You can see that one of these red chromosomes and one of the blue chromosomes have exchanged pieces with each other. That's an example of a reciprocal translocation. It's much easier to find with such chromosome painting compared with the older banding techniques. So you may see something like this on your exam. So in Chapter 3, we've looked at chromosomes, we've looked at normal structures of chromosomes, we've looked at numerical alterations of chromosomes that can cause problems for people, and we've looked at some of the common structural alterations in chromosomes also.